Good morning, everybody. Looks just like it did before the Snowmageddon, Tanya.
Did you get water or electricity, or were you out of everything?
No, we had water, but we lost electricity from Tuesday afternoon to Thursday morning. And we lost it overnight on Monday as well, and kind of on and off throughout Tuesday. So, and you?
Well, I had water every single day, so that was good. But electricity was on and off, like off for 9 hours, and then it would come back for 30 minutes. So I was just ready to get cooking. Like I had all like prepped, and I was running out of food already. So it was crazy. I was just like dicing potatoes and okay, let's just eat like potato tacos or just kind of like no eggs, no chorizo, nothing, just
Right.
simple stuff.
Yeah, we had a very full kind of pantry, all dried food and stuff. So, and we had a camping stove from, when we go camping and plenty of propane. So we're okay for that. But yeah, it got a little chilly inside. And the the kids were so bored. It was hilarious. It's like we can't do anything.
Yes, I have a 10-year-old, a 9-year-old, and a 4-year-old. So yes, they
Wow,
were like on board. So I was playing with them. I was playing Loteria and Uno, and I was like, okay, no more.
that's funny. We've got a really cool game called Rat-a-Tat-Cat, which is it's a fun card game, and because my kids are nuts about cats as well. But it's kind of a memory and accounting game. So you have to remember what cards you have and what cards your opponents have. And it's a really cool game. It's well worth, probably not for your 4-year-old, but certainly for your 9 and 10-year-olds. Although obviously they've got electricity now, so they'll be on their devices, I imagine. Uh-huh. My daughter just came down. Okay, so let's get cracking. So what we have to do today is to start on non-Mendelian inheritance patterns. Before I start, does anybody have any questions about, I guess you probably wouldn't, but any questions about assignments, homeworks, things like that before we get going?
Just to make sure, labs are gonna be the same schedule. So tomorrow's the B lab, even though they were last to go to the lab.
Correct. Yes.
Okay.
Yeah, so the whole thing's been shifted back. This should also apply to Mr. Penny's lab sections too, for anybody listening in that is in one of his lab sections. So you'll attend on the same day as you did before. All that happens is that now the order in which the different sections do it kind of flips around. So whereas, in my section, the A lab would be the first lab to do the lab for that week, and B lab would be second. Now it's going to B lab is going to be first, and A lab will be second. But you'll be attending on the exact same date. So if you're already scheduled, like work shifts or whatever for the lab period that you're not supposed to be there, that stays the same. So we're not changing which dates on which you attend. We're simply kind of pushed everything back a week, and that means now that the second half of the lab starts the lab. And the only other change is that some of the deadlines have obviously been pushed back a week as a result. So make sure you change those. They're in the schedule. They've been changed in the schedule. They've been changed in Blackboard, but make sure you change them just so that you remember when things are due. And also, the last lab session, which was supposed to be a open lab about the lab report, the mini lab report, is not going to happen. So we'll just have to deal with that. So I got to show you. I had one of my foster pups came back. Oh, there he is. This is Benji, and he's after stealing my other dog, wherever she's gone. There she is, stealing her bone from her. So we'll probably get a little bit distracted. This little pest down here starts yapping and yowling at my other dog. Yeah, so that Zoom session is gone to make space for shifting everything back 1 week. And we'll just have to deal with questions about the mini lab report kind of throughout the lab sessions as they go, and by email and the like. Also, you should have already seen the email about the lecture schedule has been changed. And as a result, the homework deadlines have been changed. They're still on Saturdays, right? So that hasn't changed. Pardon me. Hello, Kim. But the date at which like homework 3, homework 4, homework 5, and so on has changed, and it's different for each lecture section as well because of the way the things are done. So please refer to the schedule. If you ask me, I'll refer to the schedule because I can't remember either, right? They're different for both of them. So make sure you change that as well, because I don't want you to be kind of out of luck and kind of missing out on different things. Hey, Kim, sorry, I've got to show Kim talked about. Here's one of my foster puppies came back.
He's so cute.
He is. He's a little rascal, is what he
So
is.
he came back.
Yeah, while my wife was away in Atlanta for 3 weeks, another foster parent looked after him. And
Okay.
by all accounts, he was a little bit of a terror with their other dogs. And so now he's back with us until he gets adopted.
Well, that's good. That's good. He has a nice place. That is tricky with foster dogs and other dogs in the house.
Yeah, he's a little cutie. I think he just didn't like one of the other dogs for some reason. I don't know why.
Probably the same reason why we don't like some people.
Yeah, probably.
Yeah.
He's a little firecracker as well. So
He's
anyway,
cute.
I may get a little distracted every so often when these two start going at it,
That's understandable.
but we'll see. Okay, so anybody have any other questions about lecture or lab? Also, as just a quick update, all lab sections should now have the same assessments and grading structures as each other. So that has been, at least as far as I'm aware, it's been fixed. So if it appears not to have been, then let me know and I'll sort that out. Okay, so now we get into the fun part of transmission genetics, because to be honest, Mendelian genetics is a bit boring. It's really useful and it's important to learn because it gives you that foundation for understanding like the more fun genetics. But Mendelian genetics in and of itself is, it's kind of not super fun and sexy. Now, non-Mendelian genetics is where things start getting pretty weird and wacky. And so we're going to start getting into this with, let me make sure I can share the right things with you. Let's see, where are we at? Shh, Soph, teaching. All right, just reminding my daughter that I am actually teaching a class. Anyway, so this is actually, went off on a little bit of a history tangent yesterday with my other class. This is Tsar Nicholas and his family. So he was the Emperor of Russia until the Bolsheviks kicked him out and actually brutally murdered all of them in a forest in Siberia, which is a little bit rough. But anyway, this family, particularly this little champ here, Alexei, had an X-linked recessive trait, right? So a sex-linked trait, which we'll be talking later on in the lecture. And this is also an issue in the British royal family from Queen Victoria. And so someone asked me, are these two families related? And actually they are, because all of European royalty is like, interbreeding and inbreeding up the wazoo. And so he is first cousins to Queen Victoria's grandson, or was, obviously, because he's dead now. And even weirder, this is super weird. Current Queen Elizabeth II, yay, she's awesome, is married to Prince Philip, obviously. He's a bit of a crotchety old sod, but they share the same great-great-grandmother. It's like, yeah. So I always like to think that this current round of like Harry and William actually marrying people from other continents is like this huge like innovation in royalty of outbreeding. They're not marrying their cousins and their aunts and their uncles and stuff anymore. Anyway, so that's a bit of an aside. It's just fun to talk about this stuff. So what we're going to talk about first is sex determination, right? Because we have a clear physiological difference between sexes of any given species, particularly sexually reproducing species, which we call, where's the chat gone? Here we go. It's an important word for biologists, if I could spell it right, I think. Yeah, that looks about right. Is dioecious. So species that are dioecious reproduce sexually. Right? It's not about whether they're out morning or night, but how they reproduce. We're also going to talk about sex-linked inheritance, which is a clear break of Mendelian inheritance. Maybe not towards the end of the class, depending on how we go. I want to start talking about other funky stuff, but we'll definitely cover those next Tuesday. Oh, also, just as a reminder, this Thursday, crap, I've got to make myself a reminder. I hate it when I haven't used a pen for a while because it really hurts. There you go, make a reminder to do that. This coming Thursday is our last invited lecture stroke extra credit opportunity. We'll be having a microbiology virology candidate coming, and he's actually, from what I remember, because I'm on a lot of these searches, he's actually interested in extremophiles and astrobiology, which is one of my pet kind of hobbies scientifically. And so he's going to be talking about all kinds of cool stuff on, extraterrestrial planets and things, possibly. I don't know. We'll have to see. But anyway, the assignment is already posted. Where are we at? Find the right one. Here it is. So that's, Phil, let me back in. I hate the way this thing logs me off like as soon as I blink at it. So that's in the exams and quizzes, same place as before. It's here. You will need to use a different form because it's for a different search. They are different for whatever reason. And as with before, you need to attend the lecture on Thursday. I'll post the link actually like after this because I got it yesterday evening. I forgot to post it. And then upload your feedback form by Friday midnight. And if you do all that, you get your 10 points of extra credit. So it's kind of like being on a lecture, but you get paid for it in a sense. And you get to listen to something, will hopefully be really cool and novel and fun to listen to. So that's that there. So make sure you do that, and it'll give you a little bit of a boost. All right. Sorry, trying to drink my coffee before it gets cold. this lecture is right in the middle of my, regular coffee schedule. So I'm trying to figure out the best way of doing that. So essentially the idea about all of this stuff, non-Mendelian inheritance, is that, again, Mendelian inheritance patterns are whole monohybrid, dihybrid, and the laws of inheritance, laws of dominance, which is fairly straightforward, law of segregation, and law of independent assortment, they're really just kind of thought structures, right? It's like the laws of physics, right? You want to see how those are kind of bent or broken. And with this, those laws are, it's almost like they're idealistic, right? They're very prescribed, they're very useful, but they don't always apply. In fact, they fairly rarely apply, to be honest. Particularly with humans, typically the only time that Mendelian inheritance applies kind of as we've learned it is with single gene disease causing traits. So typically congenital diseases such as like Tay-Sachs, cystic fibrosis, things like that, right? Kind of autosomal recessive traits that is caused by a single gene. So most traits in most animals, particularly in humans, are not caused by one gene, and they are not necessarily a simple dominant recessive situation, right? So if you kind of just use those basic Mendelian inheritance principles to try and figure out, complex traits, you'll really struggle. So that's why we want to talk about this kind of all the exceptions, right? And really, like, the exceptions are the rule in a sense, because most traits behave in complex and funky ways, right? There are very few of them are simple Mendelian traits. And so we're going to start off with sex determination because it's kind of neat, actually. And it's also obviously a difference between of genotypes between different sexes, right? And different different phenotypes resulting from that. So got that done. Ever so quickly before we kind of get into the genetics of it, it's just this kind of basic idea that sex is not necessarily determined genetically, right? Can also be determined environmentally, right? And so the classic one is the reptiles, depending on the incubation temperature for their eggs, determines what sex those offspring end up as. Even weirder, actually, just another example occurred to me from work I did in my PhD. I'll talk about that in a second. Several fish species, they not only can change the sex of their offspring, but they can change their own sex depending on how many members of their own sex are around them, the sex ratio of the population they're in. So if you're a male fish and there are a whole ton of male fish around, then in terms of reproductive fitness, you may be better off becoming a female, right? So you're more likely to be able to pass your genes on to the next generation. And so those fish actually change sex into the opposite sex, which is crazy. It really blows my mind how they can just go, all right, and remodel their bodies as they're still alive so that they can do the other stuff. But anyway, another cool one is actually a parasite that I worked on for my PhD. It's able to determine the immune environment around it. We still don't know how, even though I finished my PhD like 15 years ago, and change the sex of its offspring as a result, so that there's more sexual reproduction to evade the immune response. That's a little bit different because they are, shoot, what's the word? Dang it. I should remember, I did this for like 5 years. They reproduce asexually. And so, or kind of, it's kind of weird. But anyway, so the idea is basically that sex is not necessarily a fixed condition in a lot of species. It's one which is conditional on the environment. Now, obviously, as you get more complex, although, you know, fish are pretty complex, they have a spine. Certainly as you get into mammals and other species, or you can have different ways of determining sex. And so just before we get into that, a little bit of language. We have autosomes, kind of like Autobots, and we have sex chromosomes, right? So autosomes are simply not sex chromosomes. They can actually play a role in sex determination. This is not entirely correct, as we'll see in a little bit. The sex chromosomes are different morphologically and differ in number or shape or size between males and females. So males and females are different in terms of the number of different chromosomes they have, or the size of them, or whatever, right? And we'll talk about some different ways in a little bit. And there are lots of different ways you can do this too, right? And there's also a few examples of convergent evolution, which is pretty interesting too. And so you're probably already familiar with XXXY, right? So we as humans use that, where females are the homogametic sex, right? They have two X's, same chromosomes. Males are heterogametic. Insects, Drosophila, those things that you've been tormenting in lab, also use this, but they use a very, very different mechanism to determine male versus female. Males are also heterogametic, XY, but they use a very, very different method to become males. Now, insects and some nematodes, they are XXXO, where O is absent, so absence of X chromosome. So in nematodes, for example, the males they simply have one X chromosome. So during sperm production, that X chromosome that sperm should inherit is destroyed. So the male gametes do not have a sex chromosome. You only get it from the female gamete. And then birds, like chickens, are also have this, they use Zs for some reason, or Zs if you're American, this ZZZW deal, but this time the males are homogametic, right? So males are ZZ, females are ZW, right? So there's lots of different ways you can do this. And it's actually, it's you know, if you're interested in kind of evolution, like seeing how these things kind of appeared, is super interesting. You know, particularly the worm that I work on now, C. elegans, is a hermaphrodite. So it produces sperm when it's younger and then keeps them in a pouch and then turns into a female and uses its own sperm to fertilize its now own eggs, which is weird. But it's a bit of an oddity because other related species don't do that. And so, you know, looking at the pathways that control that process and how they're kind of manipulated, evolutionary speaking, is really cool. So what I want to talk about next is flies, right? So this is something that I basically learn again every time I teach this class. And every time I do, I understand it a little bit better. So I'm going to teach you how I learned it, because I now finally fully understand it. How cool is that? I've only taught this class like 4 or 5 times. And as a result, I know that it's not an easy concept to get. So, you know, obviously you're hopefully not going to take this class 4 or 5 times until you get it. So what I want to do is kind of walk you through this slowly. Let's kind of put this up. So actually, you know what? I'm just going to do it all on the board. My dog is hiding from the puppy. She's up on the couch over there. There she is. She's a funny creature. Okay, so let's see if I can get this. Look at that. I spent some work. I even took a cushion out of my couch to set this up properly. Okay, so we got our irritating little Drosophila. I don't hate these as much as giant pandas, but I do pretty much hate Drosophila because they're just so irritating. Very, very cool as a, you know, a model animal and experimental organism and stuff, but super annoying if you have them in your kitchen. Right, so in, oh, actually I could even draw these. There you go, that's a female. Huh. Right, so females are XX. Oh, I've got to make that one a little bit smaller. There you go, that's a male. No, they're anatomically correct, more or less. These are XY. And so the way sex is determined in Drosophila is by counting chromosomes, right? So what we have, that's the best way of putting it. Sex equals the X to A ratio. So this is sex chromosomes. This is autosomes, right? So we're just going to use A as a shorthand for autosomes. Now this autosome count is fixed at 2. So Drosophila don't actually have very many chromosomes, which is kind of funny. This is either 2 or 1, because in terms of sex determination in fruit flies, and this is very, 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 very different from humans, the Y chromosome does not count. So the Y chromosome has some genes for like sperm production and, you know, male stuff. But in terms of sex determination, generating the sexual difference, the Y chromosome doesn't count. All we care about are how many X chromosomes do we have. And the other cool thing is that this is done at the cellular level, right? So this is done from the moment of fertilization, and every single cell counts this ratio. Very, very different to humans, which is done at an organism level, right? So sex determination in fruit flies is done at the cellular level, which is pretty nuts. So females are going to have 2 X chromosomes. They're going to have 2 autosomes. We're going to have a 2 to 2 S to A, or X to A, sorry, ratio. Males still have 2 autosomes. Actually, this laptop's super annoying because it's got the camera at the bottom of the screen, which means usually people like looking up my nostrils when I'm talking to them. But we only have 1 X chromosome. Just going to take a quick picture of that. So that seems fairly straightforward, right? Question is then, how do individual cells count how many X chromosomes we have, right? How is that ratio determined? Right, so I'm just going to get rid of this. And so we have, again, just putting this up here for kind of reminder's sake, right? And so basically what this is all about is that we have a product produced from the X chromosome called SXL. I'm going to do a small arrow here. And we have product produced from the autosome. So this is why autosomes can actually play a role in sex determination, just from what they produce in terms of gene products. And autosomes produce a repressor. And whenever you see like one of those funky T-shaped arrows, that's not because I can't draw arrows, it's because that's means inhibition in genetics speak. So again, X chromosomes produce protein called SXL, which actually stands for sex lethal. And the autosomes produce something that inhibits SXL. Just let that percolate in. It's actually really handy having people kind of on camera because it gives me a better sense of how long it takes people to write stuff down. It's a bit hard to guess when I'm just staring at myself. Okay, so what difference can you see here between females, females and males? They're going to produce the same amount of repressor. What about SXL levels?
So females have an advantage.
Right, they have more SXL produced. Right, so here's a funny question, rather than me telling you everything. What role do you think SXL has in sex determination? Do you think it's likely to be pro-male or pro-female? So you're right, Tanya, females have more SXL per unit of repressor. Males have less SXL per unit of repressor. In which sex do you think sex lethal is going to be repressed?
The males?
Correct, exactly, because there's less of it for every unit. Yeah, thank you, Adi. For every unit of repressor. So what we have is sex lethal basically is pro female. That's not freights, that's traits. And anti male traits. So if you have a 2-to-2 ratio, let's say, of SXL to rep, the repressor, this is still good. It's still able to function. It's not repressed, and it's able to do what it does, which is promote being a female and inhibit being a male. Now, if you have 1 to 2, pardon me, and just kind of yeah, that didn't work quite well. Never mind. Now, if you're males, sorry, it's not quite as spontaneous as one would hope, but it's fine. In males, you have less XX cells, so there's more repressor per unit, I guess, right? So now SXL gets turned off, which means it's no longer able to promote female traits and inhibit male traits. What you after? Oh, okay. And so you're no longer able, or the cell is no longer able to promote femaleness. And so the male traits are the default as a result. So that's how Drosophila count chromosomes. And honestly, it's taken me literally bloody years to figure out a good way of teaching that. I think last few times my students just stare at me blankly. I'm like, what the hell are you talking about? But it's really neat because it's done at every single cell, of which at the time of fertilization, technically they're not cells in Drosophila. They share a cytoplasm, but there's still hundreds of nuclei around. And so every single one of those has to independently count its chromosomes and determine whether it's male or female. Now you can actually have XXY flies, and they are what are called super females because they have no, those are super males. It's kind of weird because you can actually have more or fewer sex chromosomes in Drosophila. I don't exactly know how they do that. But essentially it depends on the ratio. So you can have XXY, and even though they have a Y chromosome, they're still female because they have two X chromosomes. And so the ratio of X to autosome is still the same. It's just super confusing. Don't ask me about that because I don't understand that fully yet. And I probably shouldn't have even mentioned it. But anyway, let's get rid of that lot. So in contrast, humans are very, very different, right? And just as with fruit flies, the Y is a very kind of stunted chromosome. It's the same in human males. I always find, I guess I'm proud of my Y chromosomes, but it's hard when they're so small and tiny and kind of almost useless. But the big difference between, so this is another kind of representation. In humans instead, this process happens quite a long time after fertilization, and it happens at the organism level, right? So in humans, until about 7 weeks post-fertilization, you are neither male nor female, which I always think is just pretty neat. So let's just write that down. And so what you have, and this is, I always think this is just a funny term. You have what are called indifferent gonads. I don't know why they fixed on indifferent as a word to describe that, because it kind of makes it sound like they don't care. But you have what are called Mullerian ducks, Mullerian, I'm not sure how that's pronounced, which are proto-female. And I think they're called Wolffian, which are proto-male ducts. Not ducks that go quack, but ducts like you have in your HVAC system. Right, so you have the potential to form either set of sexual organs, right? And so early on in mammalian development, an awful lot of organs start from simple tubular ducts, like your kidneys, for example, and kidneys, bladder, urinary tract, and actually primary sex organs kind of fall into that. All starts from this long kind of tubular structure, which basically changes and degenerates and differentiates during development. And so up to about 7 weeks, you have both, right? You have a set of ductwork, which could become ovaries and uterus, a set of ductwork, which can become testes and all the associated stuff that goes with that, right? And what happens is around that 7-week point, a region on the Y chromosome is switched on, right? And so throughout development, you have kind of clumps of genes which are turned on and off, depending on what needs doing at that point in development, right? And so this is a region which obviously isn't going to need to be turned on like in adulthood, right? Because the development of the testes is already done by that point. It needs to be turned on at that very specific time point. I don't actually know how that's controlled. It'd be an interesting thing to look into. But when it's turned on, that's the SRI region, which is a sex determining region Y. I don't know why it's called that. Perhaps they went through the rest of the alphabet before they got to that one. But essentially what it is, it's a transcription factor. And we'll talk about this later on in the course when we get to gene expression. But it's basically a protein that recognizes specific bits of DNA. And those bits of DNA will be in what are called the promoters or the regulatory regions of genes involved in testes development. And so essentially what it is a switch that once it's produced will go around and find every gene that's involved in testes development, because there's going to be a lot of them, and it will turn them all on. Right, so essentially this is a program. It's like, okay, you can almost imagine the cells like, all right, testicles are on the books for today. We need such, and kind of like turn all of those on. Then you get, you get testicles later on. So essentially once those genes are turned on, testes start developing from the Wolffian ducts, and that has two consequences. First is that the testes produce testosterone, which it's called a sex hormone, kind of irritatingly, because it does a lot of other things as well. But one of its roles is to promote male characteristics, right? So further development of the primary sex organs and then other stuff later on in life, how hairy your shoulders are, stuff like that. Funny thing is, females also produce testosterone too, right? It's not just produced by the testes. It's just the levels are different, and the other hormones present are also different, right? So that's one thing, right? So that's kind of one of those switches, right? That kind of promotes more male characteristics. pardon me, at the level it's produced at. This is also where, you know, I'm not going to get into the whole kind of gender issues because it's not something I understand well enough to teach. It's not really the right place to teach it. But obviously, if you have a hormone that produces male characteristics, but that's also produced in females, then how that level changes between individuals is going to change the extent to which they have those characteristics, right? So now you can start seeing that there's a broader kind of picture. It's not just necessarily this and this. Anyway, and the second thing which the testes produce is what's called a Mullerian inhibiting factor, or MIF. And what that does is promote the degeneration of the Mullerian ducts. And so not only do you have something which promotes maleness at the levels that it's produced, but you also produce an anti-female substance which causes those proto-female ducts to degenerate. And so those two things together make a male. And in females, which obviously don't have the Y chromosome and the SRI gene, they produce more estrogen. Males also produce estrogen, but females produce a lot more. And as a result, the Mullerian ducts don't degenerate. They develop into the ovaries and uterus. And the Wolffian ducts, because they don't get that signal to, like, you know, come on, be testicles, those degenerate over time. They're just absorbed back into the body. Isn't that nuts? Development is just like the craziest subject to think about. It really is. I could go on. I actually teach an entire course on developmental biology ever so often. Another fun thing, did you know everybody's watched Alien, right? Not Alien, Predator, Arnold Schwarzenegger, Alien versus Predator, things like that, right? Please say yes. I'm not that old. All right. You remember how Alien has like these teeth that go like that, like this kind of like scary mouth thing? Actually, when you're developing as an embryo, your entire facial structure is formed by things which come out called branchial arches. And so your jaw and nose are formed by two, your upper jaw, and your lower jaw is formed by another, I think, one. And those come together and actually fuse at the midline. Which is why, if it doesn't work properly, you can get things like cleft lip and cleft palate, right? Because those structures don't fully meet and fuse. Yeah, development is just like the craziest thing to learn about. Anyway, so you can see now that even though both flies and humans have the same XXXY, it is very complex. It's, you know, the most amazing thing is that we all end up pretty much functional, right? That's, for me, that is just astonishing, right? That you can essentially have all of these kind of choreographed decisions over a really long period of time. I mean, 9 months is not a trivial amount of time, as you probably already know. And they almost always pop out perfectly formed. You know, it's very rare that that process doesn't work. It's just astonishing. Anyway, so even though flies and humans have the same sex chromosome count and kind of system, they have very, very different means of determining sex using those differences, which is, I think, pretty neat. Anybody got any questions about any of that jazz? Well, while you do that, let me.
I have a quick question.
Uh-huh.
You mentioned that the testes secrete the testosterone to start the transformation to maleness. Is the absence of testosterone what starts the reproductive system to go the female route, or is there a substance that's secreted?
That would be estrogen.
Oh, okay.
Yeah. I don't know. I'm almost positive there's more to it than just that, because obviously females produce testosterone too. They just produce it at lower levels. I don't know if it's a timing deal. I don't know if it's a level, you know, so which is produced first matters or the relative level of the two matters. Males produce estrogen as well, I think, but just at pretty low levels. I'm not very familiar with how the female side is choreographed. I mean, something I'd like to look into. Just find the time.
Okay, thank you.
Yeah. But one of the key points is that so you can cause a developmental change, one, by actively promoting one thing and inhibiting another thing, which is what the male path is about. But you can also do it just by activating one thing and ignoring the other one. And I'm pretty sure that's how female reproductive development goes.
Yeah, that's what I was wondering, if it was more like, that.
I think so, but I'll look into it. I'll let you know what I find, because it'll be interesting to know.
Yeah, just curious.
Mm-hmm.
Thanks.
All right. So next up on the list of weird and wacky stuff to think about is sex linkage, right? So where were we? Classic one. I love looking at lineages, particularly of royal families. You should, if you ever just want to, you know, some giggles. What is it? What is his name? Charles the something. Last Habsburg king of Spain. You should look at that family tree. It's just awful. Actually, let's look at it now. Sorry, I'm kind of. Go on, get out of the way. Where is it? Last Habsburg king of Spain. Oh, there he is, Charles II. Isn't he a looker? Ha ha. You could actually, there's all kinds of fun stuff written about this guy. Basically, you know, like he couldn't speak until he was like 5 or 6, something like that. He only had one testicle, all kinds of funky stuff. Let me find here it is, that's what we want. Come on. That's on Reddit. But anyway, just look at this. It's nasty. You know, this lady married her uncle, second cousins, first cousins, go away. This one married her uncle, who wasn't exactly a looker anyway. And essentially what happened was this guy managed to make it into his mid-20s, I think, and just keeled over. That was it. He could barely even talk. Yeah, an astonishing amount of inbreeding. Like, this is a poster child of what you shouldn't do if you're a royal family, if you want it to survive. And basically, it just died out with him because he was unable to, he was infertile, essentially, amongst all of his many other issues. So yeah, again, you can just read Charles II of Spain autopsy, which is also really fun to look at. But anyway, that's another aside. So essentially what we're talking about is sex linkage is a trait which is expressed differently depending on whether you're male or female. So typically, if you have an autosomal trait, so a trait which is encoded by gene or genes on the autosomes, the inheritance of that trait will be equal, on the expression of that trait will be equal for males and females, right? There will be no difference between the sexes. For sex-linked traits, that is not true. So actually, you've got one on here, which you might, you can actually read the title in my address bar. Anybody think of other sex-linked traits in humans? I don't suffer from any of them, which is kind of nice, but my uncle suffered from one. And when you think what often happens to guys as they get older, other than getting hairier shoulders and hairs growing out their ears?
Baldness.
Yeah, male pattern baldness is a sex-linked trait.
When I
I
learned,
think, go
oh,
on.
well, I learned that it was determined by the female's side. Is that
It's
correct?
transmitted by females.
Right. And when I learned that, I was really surprised.
Hmm Color blindness is another one. That's also caused by genes which are on the X chromosome. And hemophilia is a classic one, right? And so, and hemophilia is, I think it's, I looked up once, the inability to produce thrombin, I think, which is a protein which causes blood clotting. And so if you have hemophilia, then essentially you can bleed to death from a small cut because it will not clot. Normally what happens if you cut yourself, or if you're stupid and use an angle grinder without a guard and cut into your finger, is that your blood will produce a cascade of reactions which cause proteins kind of stick together, right? Normally they're kind of like so, and they don't stick, thrombin proteins. And then once they're cleaved, they open up and they mat together to basically block the outflow of blood. If you have hemophilia, that doesn't happen, and so you just keep bleeding. And typically, hemophiliacs, or at least in olden times, didn't actually end up reproducing. So Leopold here was a bit of a rarity because, as you can see, almost all the others, we don't have a expiration date on them. But most of these males died before reaching adulthood. And so the reason why this is a trait which you only see expressed in males, right? So the dark red is where you have the hemophilia disease, and the pale pink is where you carry an allele for it. So these individuals are heterozygous for this trait. And because it's a recessive trait, if you're heterozygous, you don't, pardon me, you don't show the disease, right? You need two, if you're female, you need two alleles, right? Two recessive alleles to be hemophiliac, which is very, very rare, right? Now, if you're male, you only have one X chromosome. And so if you have a recessive allele in a gene on the X chromosome and you're a male, there's no other X chromosome to have a dominant allele to mask that recessive allele. And so if you have a recessive allele for hemophilia, right, for thrombin, I guess, and you're male, you will have hemophilia because there is no other chromosome. Because normally on autosomes, right, you've got two chromosomes. So if you have a recessive allele on one chromosome, there's a good chance you have a dominant allele on the other one. And so that recessive allele is masked. For males, if you have any recessive alleles on your X chromosome, you do not have a way to mask that. Oh, that's cool. Let's see, Olivier. Oh, that's crazy. Oh, because if she had complications in delivery, I guess, then she could potentially die from that. That's crazy. I never thought about that. It's really, really rare for a woman to have hemophilia because both of the parents, so the mother has to be a carrier and the father has to have hemophilia for a female offspring to be hemophiliac, because you have to get a mutant X from both parents, basically. Whereas if you're a male, you only have to get a mutant X from your mother. And so if you look at this lineage, you'll see you can actually figure out the genotypes actually from this, which is always, these are just like logic puzzles. They're kind of fun. So Queen Victoria, before she got all old and boring and wearing black, obviously had a bunch of kids with Prince Albert. There's actually a museum in London called the Albert and Victoria Museum, which is super cool if you're ever in London to go there. Anyway, she was a heterozygote. So she was a carrier of, oh my God. Wow. Oh, yeah. Sorry, Eddie. I don't think I could answer that question. Yeah, that's crazy. So Queen Victoria, however, did not express the phenotype. She was not hemophiliac. She was normal, right? But she carried the allele for hemophilia, right? That's why they're called carriers. And that's actually it's a common term, right? You know, if you have a history of like familial congenital diseases, like say Tay-Sachs, quite often you can be a carrier for that disease and not know that your offspring could potentially get it. That's actually one reason why quite often in Jewish marriages, they will check family history for Tay-Sachs. That's one reason why there's such great ancestry records for people of Jewish descent, I guess, religion, I'm not sure, whichever, that group. Anyway, so you can see from Queen Victoria, Alice, which is a rather nice name, she inherited a mutant X chromosome from her mother. Beatrice also did so. Leopold also inherited a mutant X chromosome from his mother, but because he's a guy, got a Y chromosome from Albert. And so as a result, he expressed the disease phenotype. He had hemophilia. Now, he can give that mutant X chromosome to a daughter. He cannot give it to a male, to a son, because he can only give a Y chromosome to his son, right? So you see, actually, we don't see if they had any other children. I don't know if they only had one kid or if he like popped his clogs after that or whatever. But Alice had that allele from him, right? So his daughter became a carrier and passed that allele on to her son, Ruprecht, who then expressed the disease, right? And you can see all through here, right, offspring of Beatrice. Unfortunately, both these guys got the mutant X chromosome. I don't think it is a case. I don't think there's any way in which hemophilia interferes with fertility of males. It's just whether you survive long enough to reproduce. And so nowadays, like in modern era, I imagine there are ways to manage hemophilia, you know, pharmacologically. I'm not sure how, but I presume that there would be. Back then, there wasn't. And so Leopold, for whatever reason, made it, and his cousins, does that be right? Cousins, I guess so. Nieces, nephews. I'm really useless at ancestry terms. Presumably didn't. So that's a pretty common trait. Typically, hemophiliacs would die by their mid-teens, simply because the odds of getting a cut, even if you knew that you were hemophiliac, which wasn't necessarily the case, but the odds of getting an injury which would result in your death through bleeding out were cumulative. So the older you get, the more likely it was that you would die from it. And so whenever you see a family tree, right, I could even give you one of these in an exam, and you see expression of a trait restricted almost entirely to the male sex, and you see females as carriers only and not expressing that trait, then that's a sex-linked, X-linked recessive trait. I think there are very few X-linked dominant traits, but this would be an X-linked recessive trait. And a classic way of finding out what a recessive traits are is typically you'll see that the expression of that trait, of that recessive phenotype, will skip generations. And so here you can, if we go through the Beatrice line, right? No disease, no disease, right? We know that the genotype is a heterozygote, but she doesn't have the disease, right? So it's not expressed. And then in the third generation, we have disease, right? So if you also, even for autosomal recessive, if you look at a family tree and you see a skipping of generations in terms of expressing that phenotype, that tells you that that phenotype was recessive, right? Because if it's dominant, you'll see it in every single generation, almost without doubt. Funny dog. Any questions about any of that? As you probably guess, I love looking through family trees. I just find them super interesting. They're like little logic puzzles. You can actually figure out genotypes all the way through based on who gets what at different stages. So it'd actually be interesting, you know, again, not enough hours in the day to trace this back to Tsar Nicholas's family and see who his son, he must have got, his son must have got it from his, yeah, from his mother, Alexei, not Alexei, can't remember what, Katerina, I think, Catherine was her name. Anyway, just to see where she got it from, right? Which side of whichever royal family she gained that from. And another way as well, which you can, hey, shush. Our foster puppy likes barking at stuff. Don't know why. Trying to train that out of him. Be a good guard dog, that's for sure. Ours wouldn't. Ours would be completely useless. She'd be like, "Yay, robbers, come in and pet me." Another way that you can figure out that something is sex linked is that you'll get different results depending on which way round you do the cross. This is called a reciprocal cross. So basically, if you have in a normal autosomal recessive trait, if you do a recessive male dominant female together, you'll get X output. And if you do a dominant male recessive female together, you'll get exactly the same output. So it doesn't matter which sex is the homozygous recessive and which is the homozygous dominant, you'll get the same output in the F2 generation. Now, if that trait is sex linked, that does not apply. So if in this case, this is white-eyed, the white gene, right, which you'll be working with in lab. So, the males or the flies have white eyes. If you start with it on this side where the male is homozygous recessive, or actually it's just recessive because there's only one X chromosome, and you cross that to a wild type female, then when you go all the way through to the F2 generation, you'll get a 3-to-1 ratio, right, of individuals that are red-eyed to individuals that are white-eyed. Now, if you do it on the other way around, right, because now we've got basically 2 mutant chromosomes that were contributing to the mix, right? This is actually one of the reasons why it's different. If you look on this side, we have 1 mutant X chromosome, right, which you can follow down to here, and then you can follow down to here and here, right? If you do it the other way around, you now have 2 mutant X chromosomes because it's from a female, right? So now you have a heterozygote and a recessive male. And so when you cross those together, now you have a 1-to-1 ratio, or 2-to-2, depending on which way you want to shake it, between red-eyed individuals and white-eyed individuals. So these ratios are different. So this is like an experimental way in which you can determine sex linkage. In humans, obviously, you know, you're not allowed to like cross people with each other and look at their babies. So you have to go by what's already happened, you know, over time in terms of linkage or lineage, sorry. With animals like fruit flies, you can do whatever you like to them. And so you can actually determine this experimentally. And that was actually one of the very first genes which was kind of mapped to a chromosome. This was by a guy called Thomas Hunt Morgan back in the early 1900s. So he's really the father of modern genetics. Pardon me. Mendel's like the grandfather essentially, but Thomas Hunt Morgan was the one who discovered the white-eyed flies and determined that that white trait was sex-linked. Right, so this is obviously a huge departure from Mendelian genetics because this shouldn't happen, but clearly it does. And we'll actually come back to Thomas Hunt Morgan when we look at linkage, because he also started working on something which kind of leads to another major break with Mendelian inheritance, which is linkage between genes. We'll get to that next week. Yeah, I think that's next week, next Thursday on the schedule. And so just ever so quickly in the last few minutes we have left, just kind of like a funky thing to talk about. I'm not going to look at the chickens because I don't think that's super useful. But the Y chromosome actually is pretty weird in many ways. It doesn't have very many genes on it. It's very small. Sure, I wish I had a picture of the haplotype up. It's like kind of like very, very much reduced. And also it doesn't go through much homologous recombination. So if you remember back to meiosis, particularly in prophase 1, where you end up with the homologous chromosomes kind of stick together, right? And then cross over like so. Y chromosome doesn't do that, right? They are kind of, the X and the Y are held together in meiosis, because otherwise you'd have all kinds of problems like Klinefelter's disease, for example, which is individuals who are XXY, and they kind of express, it's a really weird thing to look at. They're kind of male, but they're not fertile. So those are Klinefelter's. That's a case of aneuploidy. Oh, that's a cool word. I taught my my oldest daughter a new word yesterday, which was pleiotropic. Basically means one gene affects many different functions. So aneuploid is when you have the wrong number of chromosomes. So Down syndrome is a classic one, right? So that's trisomy 21, where you have 3 copies of chromosome 21. But you can also have that for the sex chromosomes as well. I can't remember what, you can have just X, like X0 essentially. I can't remember what, I think that's Edwards. I can't remember, to be honest. But that also produces, that's phenotypically male, but also infertile. So it's kind of weird anyway, talking about it. But anyway, just in the last couple of minutes, because this Y chromosome doesn't undergo much recombination, essentially what you can do is follow that Y chromosome through history, right? And so you can actually look at the combination of alleles on that Y chromosome, which is called a haplotype. Let me write that down quickly. And that's essentially like your individual barcode, I guess, right? Kind of one way of saying you are this individual versus, kind of almost like the CSI type stuff, but just for the Y chromosome. But anyway, you can actually trace the lineage of people descended from Genghis Khan, right? So Genghis Khan was a Mongol emperor who basically conquered from the Pacific Ocean all the way into the Stans. He almost made it to Turkey, right? And he was actually quite an astonishing individual, as was his son Kublai Khan. And did you know he actually invented the first check? So, you know, if you ever go, probably don't anymore, pay for your groceries with a check or write a check out for some like school activities, that process of writing a paper version of money was invented by Genghis Khan, because his empire was so huge that that was the only way to get money from one side to the other without it being stolen in between. How cool is that? Anyway, you can actually follow the descent of, and this was, was that like 1,000 years ago, I think, give or take. I can't remember the exact time of when he reigned, but it's about 1,000 years ago, give or take. You can actually determine the lineages descended from Genghis Khan. And so you can see, obviously, in Mongolia and northeastern China, you know, quarter of the males have some trace back to Genghis Khan, and that obviously is almost non-existent in areas that, pardon me, he did not control during his empire. And so this is, you can actually use these kind of traits as a really cool way of looking at how populations move over time, right? You can do the same for red-headedness, actually, which originally arose in Western Russia. I don't know how long ago, and kind of spread out from that point to Northern Europe. From a town called Irkutsk, I believe. They actually have an annual redhead celebration festival where people show up that have red hair, and everybody has red hair. Got pictures of it and stuff on the news. Anyway, so this is just another way of kind of thinking about that oddity of the Y chromosome. In and of itself, unless you're interested in kind of migration and population flows, it's like, nah, you know, whatever. But you can also do the same, and again, this is another tangent, for females, because only females, so you inherit DNA from your mother and your father, but you also inherit DNA only from your mother, which is in your mitochondria. Because mitochondria are not present in sperm. There's only enough space for the DNA in the sperm head. And so you can actually trace your lineage back tens of thousands of years via the mitochondrial DNA that was passed down through your mother and her mother and her mother and her mother, which is also kind of cool. Anyway, I better shut up now because we've gone over time. So what we'll do on Tuesday next week will be to look into other funky stuff, like incomplete dominance, codominance, and epistasis. And actually, I think I have, where would that be at? Let's see. I'll show you on Tuesday. Got to find it. My daughter did a really cool little bit of homework for me on leopard geckos. So I think I have a new example of incomplete dominance, which will be fun to talk about. So we'll do that all on next Tuesday. And don't forget, this Thursday is the invited lecture. I'll send an email around about that with the link. All right. Thank you very much, everybody. Sorry I went a little bit over time. And I'll see you definitely on Tuesday, and I'll hopefully see you in the invited lecture on Thursday, or in lab on Wednesday. Have a good day.